Okay, so let's start again. So what we're going to be doing is we're going to start out with phospholipids. We're going to put the phospholipids on the surface of the water. Their tails will point upward. Their heads will then be facing the water structure, the dipole moments. Then we're going to generate a micelle. We're going to dunk that structure underwater. But the problem with that, even though the phosphate head groups will point out to the water and the tails will point inward, is it's unstable. Put a little bit of water among those tails and it goes to bits. Then we'll generate a liposome. We're going to organize these phospholipids now in a bilayer so that the heads will face out to water and the tails will point inward toward one another. That will give us a proper bilayer and a liposome. And the major difference between a liposome and a planar bilayer is simply the dimensions. A liposome is no more than, maybe a fraction of a micrometer in diameter, whereas when we begin talking about cells, now a cell will be typically, pick a number, about 10 micrometers in diameter. So its circumference will be something like, 30 micrometers. And so now we'll see once we start putting these phospholipids in a planar in an arrangement of a cell, we'll effectively have a plane with very slight arc from one phospholipid to the next. So the phospholipid planar bilayer is going to be the basic structure of the biological membrane. So we're going to lay out a phospholipid bilayer, but what's different from the phospholipid bilayer and a proper plasma membrane is that these phospholipid bilayers are punctuated, they're decorated, if you put your phone away to appreciate it, they're decorated by transmembrane proteins. And so we're going to generate a hydrophobic barrier to ion movement, but then we're going to insert proteins across the phospholipid bilayer to generate a hydrophilic pathway. Now, in the initial description of this, we're just going to put lumps across the membrane. They're basically going to be potatoes across a phospholipid bilayer, and we won't have really much information about how these proteins are structured. So then we're going to take some time and talk about protein structure. We talk about protein structure. As soon as we talk about protein structure, you immediately think that proteins are polymers of amino acids. It turns out with biology on the planet here, there are only 20 amino acids. And those 20 amino acids make up all of life. And the 20 amino acids are distinguished one from another by something called an R group. So every amino acid has an R group, and every amino acid has an amino group, and every amino acid has a carboxylic acid group. And what we'll do initially is lay out a simple amino acid with an R group. It could be a hydrogen, it could be a methyl, it could be a 2-carbon structure. It could be any number of structures. In fact, there are 20 different structures it could be to determine the 20 different amino acids. But they'll all have an amino group and they'll all have a carboxy group. And that becomes important because for all of the 20 amino acids, we can form a peptide bond. The peptide bond occurs and the peptide bond is a covalent bond that forms between the nitrogen on the amino group of one amino acid and the carboxylic acid carbon, the carbonyl carbon of the neighboring amino acid. So now we're going to be sharing electrons between the nitrogen of one amino acid and the carbonyl carbon of the neighboring amino acid. And we can do this over and over again. We can make a string of these amino acids, selecting one of the 20 each time we do this, until we get amino acid strings that may be several thousand amino acids long. And that then will constitute the primary structure of the protein. The primary structure of the protein is going to be the amino acid sequence. So we'll talk about the sequence of amino acids in the protein. And this is important. It's important to know the amino acids that are within each of the proteins. As we organize the primary amino acid sequence, we'll find that because of the way peptide bonds form, all of these proteins, whether they're 3 amino acids long or 3,000 amino acids long, they all have a particular orientation. They have an endedness to them, such that there's an amino terminus to every protein and a carboxy terminus to every protein. And we'll be using this terminology throughout the course. There's a kind of terminology that we'll use about which end of the protein we're dealing with, whether it's toward the amino terminus or toward the carboxy terminus. And that idea will also come up when we start talking about secondary structure. So the primary structure is simply the sequence of amino acids from the amino terminus to the carboxy terminus, stuck one to the next through covalent bonds called peptide bonds. But then there's a next layer of structure we find in proteins. The next layer of structure is called secondary structure. Now, the secondary structure does not require anything from the R groups. The secondary structure is built entirely upon hydrogen bonding between the hydrogen of an amino group and the carbonyl oxygen of another amino acid. When we lay out the secondary structure of proteins, we can find that they're almost entirely in two forms. They either come as an alpha helix. This is going to be like a spiral staircase, and we'll spend some time analogizing the protein structure of an alpha helix with a spiral staircase. And we can, instead of having steps in the spiral staircase, we'll now have amino acids. And each amino acid will go up one step by 0.15 nanometers and will turn around the helix by 100 degrees. And then it'll take 3.6 amino acids to go full circle. And we'll talk a little bit about that. And then the other potential way we can get secondary structure is something known as a beta-pleated sheet. And this is kind of a zigzag of the protein. And these are usually out in the water layer. We'll find most of the alpha helix as transmembrane proteins. And we'll talk about how these are organized. We'll see the beta-pleated sheet comes in two forms. It comes as a parallel form and an antiparallel form. And I'll try to describe that a little bit. As we get started here, let me see if we can make this function as some slides. So, okay. So what we're looking at here is just a picture. This is all of the images I'm going to show you here are in fact in the ebook. And all I want you to do here is sit back and be reminded of what we've talked about. So what we're looking at here is the dipole moment on water. Again, showing this enormous oxygen atom to two much smaller hydrogen atoms. And because of electron sharing here and the electrons spending more time around the oxygen than around the hydrogens, we get the dipole moment. The negative end of the oxygen here in water and the two positive ends associated with the hydrogens. That then gives rise to hydrogen bonding within the water. We're going to talk a little bit about hydrogen bonding later today. But this is hydrogen bonding between the hydrogen of one water molecule and the oxygen of a neighboring water molecule. Now, these hydrogen bonds are precious short-lived. They only exist for picoseconds. And then they switch around, and this water molecule will find a neighbor to associate with. And as long as they're switching rapidly and moving around, the water is in a liquid state. But once these hydrogen bonds settle into once we take enough energy out of the system and the hydrogen bonds settle into a more long-term state, then we form ice crystals. And these become more or less permanent hydrogen bonds. And the colder we make it, the longer they live with those hydrogen bonds in that position. This is simply trying to describe what I showed yesterday in some crude pictures. It's showing the structure of sodium chloride, showing the negatively charged chlorides and the positively charged sodium. And these are actually drawn to scale, so you get some sense of the size of these different ions. And then in the bottom part of the bottom panel here, we're describing the chloride interacting with the dipole moments on water, forming a shell of hydration with the positive hydrogens facing the chloride, and in contrast with the sodium, the negatively charged oxygen ends of the water molecule are facing the positively charged cation, giving a shell of hydration around all of those ions, and then providing the concept of hydrophilicity. Those ions are extremely hydrophilic, and so they easily go into solution because they can interact with the dipole moments on water. And this is a figure, again, that we drew on the board yesterday, starting with our imagination of putting the sodium chloride crystal in one part of the beaker, and then allowing the random walk of the water molecules bumping up against the sodium ions and the chloride ions to distribute those ions evenly through the water column and reach an equilibrium. We then added a barrier between the two chambers. We said we have sodium chloride ions in this chamber, but not in this chamber. And then we talked about how the random walk of the ions would move them from the region of higher concentration to lower concentration until we reached an equilibrium where the probability of moving left to right was the same as moving right to left. And over here is shown the diffusion of water through water channels, where we begin with pure water on this side, and then water then moves from right to left, and because the water concentration is always higher on the right, it continuously moves to the left, diluting the ions in the left-hand compartment, but never bringing them to zero. So there's always a certain draw or pull. There's a certain osmotic pressure that pulls the water across. And we talked briefly about the hydrostatic pressure, which then would reach an equilibrium with that osmotic pressure. Then here's where we closed yesterday, was to talk about the relationship between triglycerides and phospholipids. And we talked about the importance of the dehydration synthesis process, where we took the hydroxyl, the alcohol on the glycerol, combined it with the hydrogen on the hydroxyl of the carboxylic acid, and generated water. But in the process now, we formed a new covalent bond between the carbon of the glycerol and the carbon of the carbonyl carbon of the fatty acid, and this bond was called an ester bond. But as a proper covalent bond, there's sharing of electrons in that covalent bond. It's simply known as an ester bond, and we could repeat that 3 times, generating a triglyceride. Then, in the last portion of yesterday's discussion, what we did was to take one of these triglycerides and modify it into this structure, which is a phospholipid. Here are the 2 fatty acid side chains, the same as they were on the triglyceride. They are uncharged. They are hydrophobic. But now on the 3rd carbon, rather than forming this ester bond, we now form a phosphate linkage, OPO. This phosphate linkage links the 3rd carbon of the glycerol through an oxygen, a phosphorus, and an oxygen to now a carbon out here on what is shown here as a choline, but could be an ethanolamine or it could be a serine. In all cases, this group out here is charged. And because it's charged, it's hydrophilic. And then down here is the lollipop structure showing the phosphate head group and the hydrophobic tails. Now, this next image is one that I borrowed from Molecular Biology of the Cell several years ago. Don't memorize the structure, but I like this structure because it shows the molecular organization of these major types of phospholipids. So the one on the left here, this is ethanolamine. So 2 fatty acids, here's the ester bond, now here's the phosphate bond, and here's the ethanolamine with its charge out at this end. Similarly, this is a choline with the 3 methyls out here and the charge on the nitrogen. It's a quaternary ammonia that's pretty heavily charged. And then this one is a phosphotylserine. And I'm not going to go through it in all this detail, but if you look at this structure up here, that's basically just the amino acid serine. And so we've now attached the amino acid serine through its alcohol group, through its OH, to this phosphate. And now this gives us phosphatidylserine. The one over here is one we didn't even bring up, but it's fairly common, particularly in the central nervous system. It's called sphingomyelin. And if you look at this, there are fatty acid side chains here, but it's not based on glycerol. It's actually, if you tease this apart, you'll find this is actually based on the structure of serine. I'm not going to go into that here, but you now have sphingomyelin based on serine, but it still has the same characteristics of 2 fatty acid side chains and a choline out here on the head group to give it a charge. So what's common among all of those is they have this capacity to interact with water. And this is where we're headed. This is a pretty picture. Up here is a micelle, this is a liposome, and this is a planar bilayer. So we'll start out with a liposome with all the tails facing inward and the phosphate head groups pointing outward. But I think you can recognize that if a couple of molecules of water sneak by these phosphate head groups and get in among these tails, the hydrophobic tails, the whole thing becomes unstable and falls apart. It's because these hydrophobic tails are not stable interacting with water. So the way we modify this structure is to make a liposome. And I like this figure because it shows all of these circles on the outside representing the head groups and all of these circles on the inside representing the head groups and the tails facing one another. So now, in contrast to the micelle, where if you get water in here, it's unstable, you now can imagine a well in here lined by these phosphate head groups. We can put a drop of water in here, and now that's going to be stable. The head groups will interact with the water on the inside here, and the head groups will interact with the water on the outside. And now it doesn't take much imagination to expand that liposome up in size. So if the size of this were, say 100 say it's 100 nanometers, pick a number, it's 100 nanometers, then it would have a fair amount of curvature to it. But if we expand this thing out from 100 nanometers to what's a typical cell, where a cell may have a diameter of 10 micrometers and a circumference of 30 micrometers, then the arc from one phospholipid molecule to the next is pretty small. And we end up with a phospholipid bilayer that's planar. That's the basis of a cell. In the basis of a cell, we can then think about these head groups facing the outside world and these head groups facing the salt solution on the inside of the cell. And what we'll then see is this is a pretty impervious barrier to ions, and we'll have to modify it in some way to allow ions to move from the outside of the cell to the inside of the cell. Well, with that, though, let's think a little bit about the phospholipids and pick up where we left off last time and describe the phospholipid interaction with water. So we'll take this panel and label it as phospholipid interaction with water. And in our first thought experiment, we're going to take a beaker of water and imagine that this is an absolutely flat, still layer of water. So that beneath that air-water interface will be water molecules. And they, of course, will have their dipole moments. Negative ends here and positive ends here. Now, on the surface of water, what I want you to imagine is up here we have a kind of salt shaker. Now, we can't do this in real life. But if you imagine this was a salt shaker full of phospholipids, and you imagine we take the phospholipids and shake them out on the surface of the water, how will they orient themselves? Well, they're going to orient themselves in such a way that the charged head groups will sit on the water. But the lowest order state here, the least amount of energy, is where the head groups interact with the water, but their tails point upward. So we get, theoretically, we would get a single monolayer of these phospholipids extending across the surface of the water in all dimensions, all lined up with head groups facing the dipole moments of the water, their tails facing away. But that gives us precious little insight into how we're going to use phospholipids in a biological model. So the next thing to think about is how will these phospholipids orient themselves if we dunk them under the water and put them into the water? As I just showed in the slide, in our first iteration of this, the head groups will, of course, point outward. The lowest order thermodynamic state is for those head groups to point out to the water and the charged head groups to interact with the dipole moments on the water itself. But the tails have to point inward. Now, anthropomorphically, you can say those tails don't like to interact with water. But in physical chemistry, it just says the lowest order state, the most stable state, is for the head groups to point out to the water and the tails to point inward. Now I've drawn this as a circle, but recognize it's a proper sphere. This is going to be a three-dimensional structure, and it'll be surrounded by water. These are not very common in biology in the world at all. And the reason for that is all we have to do is take a water molecule and get it to sidle in between those phosphate head groups. Once that water molecule gets in among those hydrophobic tails, then the whole thing comes apart. It goes to bits. So this is unstable. The way we gain stability is to now imagine what I'm going to label here as a liposome. And the organization of a liposome is pretty similar to the micelle, but has a really important difference. And I always have to remember to make this big enough that I can put two layers in. So there the phosphate head groups are pointing outward. The tails will point inward. But now we can make a second layer. What we can do is we can take the tails of this second set of phospholipids so that one set of phospholipids are facing the water out here, but we can drop some water into the middle of this. We can put water in the center of this, and because the phosphate head groups are pointing to the water, that's a stable structure. And the tails are facing inward, so they're hidden from the water. And so they're in a stable environment too, because they're in an environment that's hydrophobic with all those hydrophobic tails pointing one to the other in the middle of the phospholipid bilayer. Typically, when we talk about lysosomes, they're in the neighborhood of about 0.1 micrometers. But this structure can be expanded out so that we can get this same arrangement that's instead of 0.1 micrometers in diameter, maybe it's 100 times as big, 10 micrometers in diameter. And if it's 10 micrometers in diameter, pi d tells you it's going to be 30 micrometers in circumference. So we can then expand this thing out and make this a gigantic sphere. And if we make it a gigantic sphere, then if we would imagine standing and with 1 foot on this phospholipid head group and 1 foot on this phospholipid head group, it would be practically planar. It would be practically flat. It's a little bit like going outside this building, standing on the quad out there and saying the world is flat. It seems flat to us, but we know from other experiences that it's properly round. We live on a round world. And that would be true here too. If we would shrink ourselves down to these dimensions, and the diameter of one of these heads is in the neighborhood of about 1 nanometer. So it's going to take 30,000 of these 1 nanometer balls to go the circumference of a typical cell. So the amount of arc that you get from going from one phospholipid to the next is very small. So that means all of that is to tell us that we can effectively think about these phospholipids in a cell as being arranged in a planar fashion. And then here's the other plane, the other layer. And we can arbitrarily say that this is the outside, and this is the inside. I think you can see that if we were simply to put chloride ions out here, along with their counter ions, the sodium ions, that the hydrophobic tails, the hydrophobic region of this phospholipid bilayer is so hydrophobic that these highly charged chloride ions and sodium ions are not easily going to move from the outside to the inside. And yet, a fundamental idea of all of biology is the communication of ions from the outside of the cell to the inside and the inside to the outside. So we've generated here with a phospholipid bilayer a fairly impenetrable fortress, a palisade through which these ions don't readily move. So that brings us to the question, how do we make a pathway from the outside of the cell to the inside of the cell through which these ions can move? How can we generate a hydrophilic environment through which the ions can move? And the answer is just an extension of what we talked about yesterday. We're going to put a channel across this membrane. But what we're going to see is that the channel is made of protein. Now, I'm going to draw this initially as though it's a hot dog bun. And the orange hot dog bun here is meant to be a transmembrane protein. But the transmembrane protein is folded now in such a way that it provides a kind of hydrophilic pathway for ions. So there may be, for example, an orange sodium channel, and over here, a chloride channel. But for ions to move across the membrane, they need to have a hydrophilic pathway. And what we're talking about here is a hydrophilic pathway through the hydrophobic tails of the phospholipids. Now, the distance across these membranes is small, and I won't ever ask you to hold on to this number, but it's a convenient number to work with. It's the diameter, the thickness of the phospholipid bilayer is in the neighborhood of about 6 to 7 nanometers. So it's pretty thin. The diameter of each of the head groups here that we discussed a moment ago is about 1 nanometer. So remember, we're working down at a billionth of a meter. These are tiny, tiny structures. And if we were to imagine the circumference of a typical cell, you would line up 30,000 of these individual phosphate head groups to go around the circumference of the cell. So we're talking molecular distances here. So we've blithely put a protein across the phospholipid bilayer. We've blithely said that the protein has generated a kind of hydrophilic pathway, but it doesn't tell us very much about how those proteins do this job. And to understand the relationship between the proteins and the phospholipids and how they generate a channel, we now have to spend a little bit of time talking about where proteins come from, how they're organized, and what the structure is. So let's do that on the next panel. So what we're going to do here is start talking about amino acids and protein structure. Biology here on Earth is based on 20 amino acids. And the 20 amino acids are distinguished one from another by their R groups. So there are 20 different R groups to determine the 20 different amino acids. But there's a consistent structure among all of these amino acids. And it starts out here with a nitrogen and a hydrogen and a hydrogen. This is called an amino group. That nitrogen is connected to a carbon. That carbon is connected to a carbon. This carbon has a double bonded oxygen and an OH. You'll recognize this structure from discussing fatty acids yesterday. This is a carboxylic acid. We'll refer to this as the carbonyl carbon. We'll refer to this as the carbonyl oxygen. This carbon that is in between the nitrogen of the amine and the carbon of the carboxylic acid has attached to it an R group. Again, there are 20 different possibilities here. Sometimes, for example, in the form of glycine, it's only a hydrogen. Sometimes there are ring structures out here. Sometimes there are fairly long stretches of carbon-carbon bonds. I'm not going to get into that. I just want you to recognize that there will be 20 different amino acids. And these amino acids will vary. Some of them will be hydrophobic. Some of them will be charged. Some of them will be hydrophilic in terms of having polar structures. So we can vary the structure of each of the amino acids by its R group. But having said that, the important point here is that there's an amino group and a carboxy group. And what we're going to do now is use the chemistry of the carboxylic acid and the chemistry of the nitrogen to be able to link these amino acids in great long polymers. So what we'll do next is in orange here, there's a nitrogen, a hydrogen, a hydrogen. So there's the amine linked to a carbon. We'll just put an R group up here. It could be any of 20 different R groups. A carbon, a double bonded oxygen, and an OH. We're going to spend a lot of time in showing how this happens in biology. It turns out that ribosomes are going to be the enzymes that catalyze the formation of the next bond. But what happens is that we'll take the hydrogen from this nitrogen, and we'll take the hydroxyl from the carboxylic acid and generate water. This is another great example of a dehydration synthesis. We're going to take a water out of these two amino acids, a hydrogen from this amino acid, the hydroxyl from the carbonyl carbon, generate water, and in the process, we're going to generate a new bond. We're going to generate a bond here between the carbonyl carbon and the nitrogen and the amine, and this bond is said to be a peptide bond. We can do this over and over again. We can take another amino acid here. Use a ribosome to do the dehydration synthesis. Generate the bond between the carbonyl carbon and the nitrogen. That's another peptide bond. We can do this out here again. NH, H, C, R, C, double bonded O, OH. Form another peptide bond, and I'll do this one more time. So again, a peptide bond here between the nitrogen and the carbonyl carbon, H, carbon, R, C. We can do this over and over again. Now we've shown the example here of 1, 2, 3, 4, 5 amino acids. It would be called a pentapeptide. What I want you to recognize from this, though, is that it doesn't matter whether we have 5 amino acids, or even 2 amino acids, or 5,000 amino acids. There's a kind of orientation to this molecule that derives from the formation of these peptide bonds. And that is, these protein molecules will always have, at this end, an amino terminus, and at this end, a carboxy terminus. This endedness of the molecule provides us with a kind of language. It gives us a kind of nomenclature to use when we talk about proteins. When we talk about proteins, we'll frequently refer to the amino terminus of the protein or the carboxy terminus of the protein. We'll frequently talk about toward the carboxy terminal or toward the amino terminal. So this is a common nomenclature that we use over and over again to describe proteins. So they do properly have an endedness. The other point to make from this crude drawing is that this is said to be the primary amino acid sequence. So that at each of these peptide bonds, we can now link a new amino acid. We can choose any one of the 20. And when we put the next amino acid in, we can choose any one of the 20. Now, we'll find out a little bit later that the determination of which amino acid we put in is going to be based on a 3-base set in the messenger RNA. But we'll leave that till later. All I want to say now is that we're forming these peptide bonds, and that each time we form a peptide bond, we can choose a novel and new amino acid from among the 20. Yes?
When the ribosomes are constructed proteins, are they constructed from the amino terminus to the carboxy terminus?
It's a great question. The question is, when we're generating a protein by using a ribosome, do we always construct from the amino end to the carboxy end? And that's absolutely right. We'll generate the first amino acid produced, the first amino acid in a protein produced by a ribosome will always be at the amino terminus. And that will actually become important when we start threading these proteins through the ribosome. Anything else? So now we have this sequence of amino acids. We say this is the primary amino acid sequence, and this is important because it tells us all of the amino acids in a protein, and it tells us the order that these amino acids come in as they reach from the amino terminus to the carboxy terminus. But that tells us very little about the function of these proteins. And the protein function is derived from taking this primary amino acid sequence and now imposing upon the primary amino acid sequence first a secondary structure and finally a tertiary structure. So we'll see that the primary structure is simply the sequence. The secondary structure, as I mentioned in the introduction, has to do with hydrogen bonding within the molecule. And finally, the tertiary structure will be based upon the R groups and the R group's interaction with water. And the R group's interaction with water in tertiary structure will determine its three-dimensional shape. And an important concept we'll come back to over and over again is the three-dimensional shape of the protein determines its function. So if all life on the planet, all enzymes, all activity is based on the three-dimensional structure of proteins, we need to spend a little bit of time talking about how that structure is generated. So we're going to walk through first the primary amino acid structure, meaning the primary structure, the primary amino acid sequence, and next the secondary structure, and then we'll get into the tertiary structure probably tomorrow or probably Monday. So let's take the upper panel here. And we'll label this as secondary protein structure. And secondary protein structure comes effectively in two flavors. It comes in a spiral staircase flavor that we talk about as an alpha helix, and it comes in a planar arrangement called a beta pleated sheet. The alpha helical structure, we'll see a little later, probably on Monday, is the structure that predominates in the regions of the protein that span the phospholipid bilayer. The structure, the alpha helical structure, is not particularly stable in a water environment, and so it is then either tucked in around other parts of the protein or more commonly reaches across the hydrophilic region of the phospholipid bilayer. But let's first talk about secondary structure and think about the alpha helix. So protein secondary structure. And we'll first describe this in terms of an alpha helix. Now I'll try to draw a helix on the board. It's going to be pretty crude, but I'll try. The alpha helix, and we can think about the amino terminus down here, and the alpha helix now becomes a spiral. That spiral is now constructed of amino acids linked, just as we saw above, by peptide bonds. So within the spiral, and I'm going to put circles here to represent the amino acids. So there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Now, don't memorize these numbers, but I need the numbers to walk through with you how these are oriented. So that each amino acid is 0.15 nanometers from each of its neighbors. Because this is a helix, each amino acid twists by 100 degrees. I'll remind you that it takes 360 degrees to go around a full circle. So that means it's going to require 3.6 amino acids to traverse, do it that way, a full circle. And the fourth point here is that there's hydrogen bonding. Hydrogen bonding between the amino hydrogen and the carbonyl oxygen in a pattern so-called I+4. Now, what that means is that if we say this is number one, this is the number one amino acid. I+4 says that there's going to be hydrogen bonding between the amine on this one and the carboxylic acid oxygen on I+4. 1, 2, 3, 4, 5. So this is number 5. So that means the amine here, with its positive charge, is going to be interacting with the negative charge on the carbonyl oxygen 5 amino acids up the line. 2 is now going to interact with 6. 3 is going to interact with 7. So that there's this pattern of I+4 of the amine positive charge interacting with the polar negative charge on the carbonyl oxygen. And this repeats itself over and over again. It's always every I+4, every fourth amino acid. Now let's see if we can compare this to and bring this sort of into a real world state. Now let's imagine, rather than this being amino acids in a helix, imagine it as a spiral staircase. So if you can envision in your mind's eye a spiral staircase, you know that you go up a spiral staircase, you go around a circle, usually there's a central pole. And you can imagine you walk around the spiral staircase and you go around a circle, and as you go around the circle, you go up toward the second floor. Well, we're going to do the same thing here. It's going to be a little weird though, because the steps are going to be ginormous. But if you imagine the way we've laid this out as moving by 100 degrees, and we imagine that this first amino acid, that amino acid is effectively facing this wall. But now, to go to the next amino acid, we go up a step by 0.15 nanometers, but we turn by 100 degrees. So if I were to face out into the audience, that's 90 degrees, so go a little more. Then the next one is also another 100 degrees. So we come back around to this way and a little more. And then the next one, we come around to 90 degrees and a little more, and it takes 3.6 of these to come full circle. But recognize we don't have a fraction of an amino acid. So what we're doing here is we have every fourth amino acid, I plus 4, is now going clear around the circle and being hydrogen bonded to its neighbor down the spiral staircase. So again, imagine how each of these amino acids is a step on a spiral staircase. Now normally, I think it takes about 12 or 13 steps to go a full circle on a typical spiral staircase. These are much bigger steps, because you only take 3.5 steps, basically, to go clear around a circle. Yes?
Why 100 degrees?
So why do we end up with 100 degrees? And why don't we have it arranged exactly so we go 360 degree every fifth or every fourth? And the answer has to do with the bond distances within the protein. So the only way we can get this to work with those bond distances is to twist it a slightly more than 90 degrees for each one. So we go around the circuit 360 degrees every 5 amino acids. With that, I'll let you go, and we'll pick up beta pleated sheet next time.